Good morning, dear second year students. Today we are going to discussing about conjugation mechanism of conjugation process. What is conjugation? Conjugation is the transfer of genetic material between bacterial cells of opposite mating types that are in the physical contact with each other, or a process in which DNA is transferred from a bacterial donor cell to recipient cell by cell to cell contact. Historical background. Historical background about conjugation. Discovery. The conjugation mechanism was first invented by Joshua Lederberg and Edward Tatum in 1946. They experimented with two auxotrophic strains. Auxotrophic means self-sufficient producers. Auxotrophic strains of E. coli K12 denoted by strain A and strain B. Strain A and strain B were plated on minimal medium and incubated overnight. No growth observed. Also, strain A and B were mixed together and when plated on minimal medium resulted in prototropes. Prototropes also self-sufficient producers. Mechanism of conjugation. Donor cell produces a pilus. Pilus attaches to recipient cell and brings the two cells together. Only single strand of plasmid is then transferred to the recipient cell. Both the cells synthesis a complementary strand to produce double-stranded circular plasmid and also produce pilus. Both cells are now viable donor. Discovery of fertility factor F factor. In 1953, William has determined that the genetic transfer occurred in one direction. One cell acts as donor and the other cell acts as the recipient. Cells that can donate the DNA to other cells are F plus male cells. Cells that can receive the DNA from other cells are F minus female cells. Here observing the experiment about conjugation. Escherichia coli K12 auxotrophic strain A. This is the left side one. Right side one is Escherichia coli K12 autotrophic strain B. This is the strain A and strain B. Strain A can grow on minimal medium only it's supplemented with methionine and biotin. Strain B can grow on minimal medium only it's supplemented with threonine, leucine and thiamine. The auxotropes strains required for amino acids. Mix the two strains together overnight incubation. Mixed culture of strain A and strain B. In the result, plating on minimal medium overnight incubation, growths of prototropes, growth of bacterial colonies on minimal medium. Here observing the structures of conjugation process, the mechanism of conjugation process. Chromosomal DNA, F plasmid donor, it is the donor cell. The other one is recipient cell. Recipient cell contains chromosomal DNA. Donor cell contains chromosomal DNA and also F plasmids. The F plasmids and chromosomal DNA cells contains pilus. This is the donor is male cell and the recipient cell is female. The female cells also contain female cells is called F minus cell and male cells is called F plus cells. In the second one, the pilus between the pilus attached with the female cells. This is the transfer of DNA from one cell to another cell. Next one. In the third one, relaxosomes transfersomes and also DNA polymerase enzyme. In the observation, the third part, we are observing the third part, the cell, the DNA genetic material transform from one cell to another cell, donor to recipient. Fourth one is old donor, and the separate the plus F plasmid and separate the new donor contains chromosomal DNA, and F plasmid, and old donor contains F plasmid and also chromosomal DNA. The genetic material transfer from pillars. Conjugation requires physical contact. The evidence for cell-to-cell contact was provided by Bernard Davis in 1950. The two arms of the U-tube are separated by filter. On the right side is medium containing axotrophic strain A, while on the left side is medium containing axotrophic strain B. The filter allows only the medium to move to end and fro, but not the cells on either side. When culture was plated from both sides on minimal medium, no prototrope growth was observed as in Lederberg and Tatum's experiment. Thank you.